All right, here we go, AP Bio folks. We're going to jump into kind of chapter 20 now, which is the biotech one. This is where it'll be a little interesting. This is where we have a lot of activities and labs. And so I think I can probably, well, I know I can modify stuff for you to do the activities at home and send you that way. The labs, I'll probably be filming those on my own and showing you the data. Unfortunately, it'd be better if it was hands-on, but we'll see what happens and go from there. This is really the last section of the AP exam. And really, there's a couple big things about DNA sequencing and PCR that we're going to look at. But anyhow, we've got to start out. And, you know, the front picture here with the virus and retrovirus, I just had to put it on there because I forgot yesterday. And with our virus stuff. And of course, it's just pretty cool stuff. Anyhow, let's get an idea here of what we're talking about. Oops, skip back. In 2001 was when they finally, the Human Genome Project, sequenced the 3 billion base pairs of humans. And we're not going to spend hours on it. That was a significant accomplishment. It was only, I believe your book reminds us, the fourth eukaryote to have been done in a very complex situation. And we won't get long into the whole idea of all this belief that many great things were going to come out of it and all the answers were going to be there. They weren't. To date, just so you have an idea, almost 51,000 genomes have been, oops, I think I have a number missing there. Anyhow, yeah, I think I have a number. I'll get to that number again, but well, it says 51,000. 11,000, that's correct. 11,000 are prokaryotes. I think I'm missing a zero in my genomes. About 11,000 are eukaryotes, about 250,000 are prokaryotes, and then about another 35,000 viruses. Then there's some other plasmids and things along there that have been done also. Anyhow, the idea is that since 2001, in 20 years, actually within the next 10 years, by around 2010, they had 8,000 rolling. And so if you think in about 20 years, how many genomes they have sequenced, it's rather significant. Anyhow, it has always depended on having the technology to get that far. And starting with remaking recombinant DNA, and this was on a quiz that you did because it kind of filters in and out of stuff that we had talked about, but recombinant DNA is getting a nucleotide from two different sources in the DNA, often two different species, combined in vitro, meaning in the test tube in the same DNA molecule. Okay. So, understanding of viral genetics, and this is why we looked at it, with advances in DNA technology have led to rapid increase in genomic knowledge. Now we're going to talk about plasmids and stuff with how do you grow DNA, quote unquote, grow it. How do you get a bunch of copies of the gene that you want? We'll talk about plasmids like you did with your pGlow, because plasmids are a great vector. Vector meaning something that will carry the DNA into the bacterial cell and allow it to get incorporated. Viruses, they would love it if viruses could become a vector, but they're not as easy to control as bacteria. And so we may discuss that when we have time later. Anyhow, those methods for making recombinant DNA are central to genetic engineering, the direct manipulation, okay, and then being able to use that knowledge. So for an example, on the very first part of that chapter, you can open your book and look at figure 20.1, and it shows you a microarray. And that's a great way to test different cells and stuff for cancerous genes or possibility of cancerous genes. Anyhow, let's make sure that we're all on the same page, and this list would be expanding all the time. What types of technology, where have we grown in using uh, technology stuff? Well, paternity testing, or really now any of that 23andMe kind of background stuff, certainly in solving crimes. And even now it's being used not just to solve crimes, but to determine that maybe they put the wrong guy behind bars. Um, GMOs you've heard of for agricultural purposes and stuff. Okay. Manufacturing proteins like insulin, blood clotting factors, proteins, growth hormone type stuff, um, um, gene identification, such as what mutations might be there. Anyhow, in order to use for all that stuff, if you know, what are they testing in your 23andMe? You need to make copies. You need to clone DNA, okay? And you need to work with specific genes. And that's where plasmids come in. Think about what we talked about with transcription and translation. You've got a little bit of a needle in a haystack situation here, because what you're thinking of is, with the way eukaryotic regulation is processed at every level, from the way the chromosome is folded all through the way the proteins are made, and everything we looked at with transcription and translation, you know, not a particular gene that you want to utilize or clone could be, you know, a really small percentage of the total genome. So anyhow, most methods share general features such as use of bacteria and their plasmids. Plasmids, we've talked about, are small circular DNA molecules that replicate separately from the bacteria chromosome. Clone genes are useful in making copies of a particular gene and producing a protein product, like making insulin. Okay. So what do? As you did with your piglo, foreign DNA is inserted into the plasmid, and the recombinant plasmid then is inserted into the bacterial cell. And then as the bacteria reproduces, you clone a bunch of the foreign DNA. Okay, so you can get a whole bunch of copies of your single gene. Okay. So understanding that role of the enzymes in DNA and protein synthesis was key. All right. And so this picture, I think I stole this from you. This is the big overall picture, but you can kind of see, and this is more of an in vivo thing, but you can kind of see you've got the plasmid from the bacteria. You isolate a gene of interest from a cell of whatever organism you're looking for. You get that inserted into the plasmid, put that into the cell, big growth, and then you can use it for various different technologies. Okay, here's the smaller blow-up picture, okay, that you can see here. And this is the area that becomes difficult. This is an area where virus and bacteriophages were huge, and this is where restriction enzymes came from. How do you get that inserted into the plasmid? You have to cut it and hopefully complementary base pair. But anyhow, we've isolated the gene we want. Okay. We've inserted in the plasmid, we put that into the bacteria, and then again, the bacteria just grows, and now you have a bunch of copies of that gene that you want. You can take copies of that gene and do research, or insert the gene for resistance in plants. What seems like a billion years ago, before I was teaching, when I was just gening and stuff, I worked in a plant lab, and if you like plants, great. I don't. The genetic research was outstanding. The plant and the whole idea was boring. Anyhow, we were doing a plasmid insertion for pest resistance. If you ever learn anything about plants, there's an area of the country called the Rust Belt, a little farther down south, and rust is just a plant fungus type pathogen that kills the kills the plants and has been very devastating. Different types of rust have been very devastating to crops, not just in the U.S., but over history worldwide. And some plants have that resistance and some don't. So what we were doing was isolating the resistance from, I believe it was a tobacco plant, or we were inserting it in a tobacco plant. It's been many years. But we were isolating that resistant gene and trying to insert it into a plant that didn't have that gene to see if we could make it resistant. Now, if that was a food product, that would be a genetically modified food. Would that do anything to you? That's the question of GMOs, is if you genetically modify plants or animals to make them resistant to stuff, then by consuming that, are you going to somehow find a way to incorporate that resistance or have some ill genetic effects? That's a ethical discussion for after the test. Anyhow, in order to do that, and this is why the whole virus and bacteriophage machinery came into being, you use restriction enzymes to make that recombinant DNA, because you not only want to excise the gene of interest that you want, but you also want to be able to insert it into the plasmid and then get that into the bacteria. Okay, so cut DNA molecules at specific DNA sequences called restriction sites. When did we talk about this with viruses? Remember that bacteria, as their immunity, as their ability to stop viral infections, have enzymes that will break down the pathogen viral DNA, or to some extent RNA, that could get inserted. There's about 3,000 different restriction enzymes currently produced, different names of BAMH1, HIN3, all sorts of stuff. Okay, but it makes cuts at specific nucleotide sequence series, and that yields little restriction fragments as it cuts up the DNA. Now, I believe I have the picture here. You have a couple of options. Okay. Yep. This is where I'm at. You can, as you can see by the stuff on the left, is cut here. You can see there's a GAA-TTC, GAA-TTC complementary base pairing, and you can cut between that first G and the A. Okay. Same here with the GAC-GTC, right? Or GA, whatever. Or in this case, you can cut between a series of guanines and cytosines. The left two are your best bet, because you leave what are called sticky ends. Okay, you're not blunt here. The question, if you're blunt here, is how are they going to complementary base pair? Now, you'll bring in DNA ligase, as we saw with DNA reproduction and stuff before, to bind the sugar phosphate backbone here. But what are you going to complementary base pair? Where here you can complementary base pair. So those 3,000 restriction enzymes are quite specific, and you want those sticky ends to complement the base pairing. So you're matching those restriction enzymes to not only the gene of interest, the plasmid, but the gene of interest, so you can cut out exactly what you want and have those complementary base pairs match up. Okay, so sticky ends can bond with complementary sticky ends, and then you use DNA ligase to seal the bond. Okay, so here's the example. There's your restriction site that you wanted, where the enzyme's going to cut between the G and the A's, and you get two sticky ends. Now notice that your gene of interest has the same complementary base pairs, so it can go in. That is certainly one possibility. When you are using restriction enzymes, it's very possible that you get many different fragments, but only one may have the gene that you want. And that's why you do specific plating like you did with pea glow to only have that, so you can grow those colonies that you know are the ones with the gene of interest. Okay, so anyhow, there's a recombinant DNA molecule. All right, so cloning a eukaryotic gene in a bacterial plasmid, because you certainly can. The plasmid is called a cloning vector. Okay, a vector is a DNA molecule that can carry foreign DNA into a host cell and replicate there. So plasmids are great. They try to do things with viruses, but they're not as easy to control. You want easy to obtain, manipulate. Bacteria are great. Control them. They reproduce rapidly. And the other problem with vectors is the reproduction of the cell and going lysogenic. So anyhow, as I said, yeah, isolating those genes are difficult. Plasmid isolation is that one method. And so now I look to, you can go, there's a panel in your book on cloning genes and bacterial plasmids with the hummingbird, okay? So the idea is, what if I wanted to isolate the hummingbird beta globin gene that breaks down lactose and see its function, okay? Well, here's what we do. We isolate a plasmid, an appropriate plasmid, and the appropriate DNA gene from the hummingbird, cut them with the same restriction enzyme, throw them in there, okay, add the DNA ligase, okay, so that once the sticky ends go together, you can bind it together. And then the idea is isolate the gene that encodes. Now, you're going to get many, as you can see, and you'll look in the panel, and I think I have the picture coming up. You're going to get many different segments of gene when you throw in a restriction enzyme. So that is when you want to make sure that you can also isolate the, like here, the beta globin gene. You want to set it up so you know whether it's able to break down the lactose or not, okay? So now we'll get to the picture here. Some recombinant plasmids will now have the hummingbird DNA. The DNA is added to bacteria. They grow up a bunch. Plate them on a type of agar that selects for the bacteria with just the recombinant plasmids that you want, and then you cloned it. So let's go to the picture. So here we're isolating. Okay, get your plasmid of interest. That's going to have the same sticky ends. This orange is ampicillin resistant, just like we did with pea glow, so that you can make sure that you're isolating the appropriate bacteria. Here's the LAC-Z gene that breaks down the lactose. Okay. Go back. Cut both with the same enzyme. Okay. So both the plasmid and the LAC-Z. Okay. You see here, your gene of interest is this one, but it also cut out different fragments from the cell DNA. Okay. Mix them all up, join, some will join by base pairing and seal up with DNA ligase. You can see here that you got a number of different recombinant plasmids. One that was non-recombinant because it didn't pick up any fragments and didn't get cut with the restriction enzyme. The only one you want is this one. Okay. These have other fragments, but your gene of interest is here. Okay. Mix them up, grow them up, bacteria carrying the plasmids, get them inserted into the bacteria. Okay. And then plate on the agar with the X-gal and the ampicillin. What's the X-gal? The X-gal is a synthetic deal. You can read about it in your book, but the X-gal is something that as it's broken down, it will turn blue. Okay. And so along with the lactose. Anyhow, Sorry, brain fart there. That's not what you want. The colonies of growth you want are the ones that don't turn blue, because that's got your recombinant plasmid where you inserted into the operons and disrupted the LAC-Z gene and breaking it down. Now you know you've got the ones that have the hummingbird gene in it, okay? All right. That's the basic idea. And again, your book panel explains that in detail also. So let's think about that for a minute as we wrap up. Going back to the P-glow, we discussed it, but you were doing a plasmid insertion of recombinant genes. What were our genes of interest? Okay. You may want to go back to the little circular plasmid we showed. What were we trying to change? Well, we were trying to make the bacteria glow. Where should it have been able to glow? And again, think back to the operon that we inserted the gene into. And then here's a bigger question. Why do bacteria have plasmids? Okay. That's more of a research. Why do bacteria have plasmids? Big question, because really that's what biotech was all about, simulating living organisms. Anyhow, that's a quick look at it. We're going to do some activities and stuff with this information, at least setting up for labs. Eventually, we'll talk about cloning DNA through PCR and how do you read the sequences. But this is a good start. Again, I'll have a little quiz with it to do as homework. And we'll go from there and try and figure out ways to get you activities. Anyhow, have a good day. Keep to a routine. Get outside and enjoy some of the spring weather while it's hitting. And we'll see you tomorrow.